Good morning everyone. Today we are going to discuss about the basics of immunity, that is introduction to immune system. What is the definition of immunity? Human body has the ability to resist almost all types of organisms or toxins that tend to damage the tissues and organs. This capability is called immunity. So you have to remember these definitions. Once you get the question from immunity, maybe cell mediated immunity or humoral immunity, you have to begin the answer with the definition of immunity. So you have to remember this definition. Now, what are the types of immunity? It is classified into innate immunity and acquired immunity. What is innate immunity? Innate immunity is the one that is present at birth. So it is known as inborn immunity. It's also called natural or native immunity. It is an invariable hereditary response and it is present throughout our life. It is the first line defense against the invading organisms. So whenever a pathogen enters your body, your innate immunity will try to fight against the infectious agents. Once they fail to do so, they will activate the acquired immunity and acquired immune system comes into action. How this acquired immunity is getting activated? The cells of the innate immunity will produce cytokines. So cytokines are hormone-like substances that helps in cell-to-cell communication or cell signaling. So with the help of these cytokines released from the cells of innate immune system, they activate the acquired immune system. The innate immunity is independent of previous exposure to disease-causing agents or foreign substances because they don't have any specificity in destroying the pathogen. Whichever pathogen enters your body, they try to destroy them. So there is no specificity or there is no memory involved in the innate immunity. Its efficacy improves when a pathogenic organism enters the body. There is no specificity, no memory. It's also called non-specific immunity because there is no specificity or specific mechanism in destroying a particular pathogen. So all the pathogens are destroyed in the same way. General destruction is taking place in innate immunity. The cells that mediate innate immunity are neutrophils, macrophages and natural killer cells. So natural killer cells are a type of lymphocyte. They comes under innate immunity. Epithelial cells and endothelial cells also contribute to innate immune response. They kill the pathogen directly by phagocytosis or via the complement system. So what are these complement system? Complements are certain proteins which are present in the plasma among the plasma proteins. So they are released in the inactive form. They can be activated by various mechanisms. So once they are activated, that will initiate a cascade of reactions which results in the formation of certain byproducts that will destroy the pathogen. They also produce cytokines that activate the acquired immune system. What are the components of innate immunity? Phagocytosis of bacteria and other invaders by WBC and cells of tissue macrophage system comes under innate immunity. Then destruction of swallowed organism by the acid that is present in the stomach and the digestive enzyme, that is also a part of innate immunity. Resistance of skin to invasion by organisms. Then urinary tract is protected from infections by the washing action of the urine. This is also a part of innate immunity. Presence of cells and chemicals in the blood that attach to the foreign organisms and toxins and they will destroy them. Examples are lysozyme that is seen in saliva, tears, etc. They attacks the bacteria and causes them to dissolute. So they will destroy the cell wall of the bacteria and they cause destruction of them. Then complement complex, as I have already mentioned, they destroy the bacteria. Natural killer cells are a type of lymphocytes. They destroy foreign cells, infected cells and tumor cells. So these are the components of innate immunity and all these things will be present in your body when you are born. We'll move on to the acquired immunity. So acquired immunity is also known as adaptive immunity or specific immunity. Human body has the ability to develop powerful specific immunity when attacked by microorganism. It does not develop until the body is invaded by a foreign organism or a toxin for the first time, because it is not present in your body since you are born. It is acquired gradually when you grow. And you have to understand, it starts to develop when your body is infected by a pathogen for the first time. The body recognizes these agents by specific proteins or polysaccharides in their makeup. That is, the pathogen will be having proteins or polysaccharides in the structure, and small fragments of these proteins or polysaccharides are known as antigens. Moving on to types of acquired immunity. Acquired immunity is divided into cell-mediated immunity and humoral immunity. So what is the cell-mediated immunity? Cell-mediated immunity or T-cell immunity is mediated by activated T lymphocytes. They encounter and destroy the organisms which enter the body. They help in the defense against infections by virus, fungus, and few bacteria like tubercle bacilli. Delayed allergic reactions and rejection of the transplanted tissues, they are due to cell-mediated immunity. Humoral immunity is mediated by activation of B lymphocytes, so they are known as B cell immunity. They secrete antibodies into the blood and lymph, and they are important in defense against bacterial and viral infections. Your acquired immunity is again divided into active and passive. So active is further divided into natural active immunity and artificial active immunity, and passive is also further divided into natural passive immunity and artificial passive immunity. So what is this active immunity? Resistance developed as a result of an antigenic stimulus. That is, the pathogen should enter your body and there should be active functioning of the immune system of that person. So that is active immunity. It is more effective and gives better protection. What is natural active immunity? Due to clinical or subclinical infection by a microbe. Example, a person who has recovered from measles, they develops this natural active immunity. What is artificial active immunity? That is induced by vaccines. You know that in active immunity there is active participation of the person or the host. So vaccines are preparations of live which is inactivated or killed microorganisms or their products used for immunization. So what is happening? We will be injecting the live inactivated or killed microorganisms into the body, so they cannot create any disorders, but they can induce the formation of antibodies by the active participation or processing by the immune system. So these antibodies will be stored there. So when this same pathogen enters the body for the second time, initially it was injected as vaccine. It was inactivated or killed. But later, when it enters the body for the second time, these stored antibodies will go and destroy them. So that is the purpose of vaccines. Anyways, in vaccines there is involvement of the immune system in providing or producing antibodies which will be stored for the future purpose. So that is an example of artificial active immunity. What is passive immunity? It is transmitted passively to our recipient in a ready-made form. That means our immune system doesn't play any role. There is no active participation of our immune system. No antigenic stimulus, preformed antibodies are given. So it is less effective and it is inferior to active immunity. But there is an advantage. What is the advantage? It acts immediately. See, when your body is processing and when your immune system is responding and producing the antibodies, means it will take time. But here there is no role for your immune system. We are giving the antibodies in a ready-made form. So it won't take much time. So that can be employed when instant immunity is decided. So the advantage is it acts immediately. Now, natural passive immunity. So you understood what is passive immunity? That is ready-made antibodies are given. We give ready-made antibodies, so there is no active participation of your immune system. So natural passive immunity means transfer of antibodies from mother to fetus through placenta. Also, transfer of IgA antibodies through colostrum from mother to the child. In both this mechanism, the immune system of the child is not taking place. It is not doing any job. Artificial passive immunity. The resistance passively transferred to a recipient by the administration of antibodies, activated T lymphocytes, or both, obtained from the blood of someone else or some animals that has been immunized against the antigen. So these preformed antibodies or activated T lymphocytes or both are injected into the person. So that is artificial passive immunity, and the examples are tetanus immunoglobulins, rabies immunoglobulins. Moving on to development of immune system. Both the T lymphocytes and B lymphocytes, they are derived in the embryo from pluripotent hematopoietic stem cells. All these T lymphocytes and B lymphocytes finally, after their final maturation, they are dislodged in the lymphoid tissue through the blood. So before doing so, they are further differentiated or pre-processed in the following ways. Thymus pre-processing of T lymphocytes, that is, the processing of T lymphocytes takes place in the thymus. So the T lymphocytes that is synthesized in the bone marrow, they will be released into the blood circulation. But that T lymphocyte will not be mature enough to do their job. So they have to be further differentiated or processed to make them mature to do their function, and that happens in the thymus. So those T lymphocytes released from the bone marrow can be called as lymphocyte precursors because they are not matured. So these lymphocyte precursors, through the blood circulation, they reach the thymus where they divide rapidly. Different types of thymic lymphocytes with specific reactivities against different antigens develops. That is, one lymphocyte develop specific reactivity against one antigen. Then the next lymphocyte develop specific reactivity against the next antigen. That is, a single lymphocyte cannot react against two different antigens. One lymphocyte will be always specific to one antigen. This process continues until there are thousands of different types of lymphocytes with specific reactivity against thousands of different antigens. Thymus selects lymphocytes to be released by first mixing them with the self-antigens from body's own tissue. So if that lymphocyte reacts with the self-antigen, it is destroyed and phagocytosed. And this happens to 90% of the cells. Only the 10% of the cells are found without reacting to the self-antigen. The cells released, they act only against antigen from outside sources like bacteria, toxin or transplanted tissue. Pre-processing occurs shortly before birth or for a few months after birth. These pre-processed lymphocytes, they leave the thymus and lodge in the lymphoid tissue by blood. So the lymphoid tissue like lymph nodes, spleen, etc. Removal of thymus several months before birth prevents the development of cell-mediated immunity because that final maturation doesn't take place. So if this is done, transplanted tissue has less chance of rejection because we have already studied that the rejection of the transplanted tissue is done by the cell-mediated immunity. The cell-mediated immunity is mainly activated by the mature T cells. If there is no thymus, there is no maturation of the T cells, so there is no cell-mediated immunity. If there is no cell-mediated immunity, there is no rejection of the transplanted tissue. Moving on to pre-processing of B lymphocytes. So T lymphocytes, after synthesized from the bone marrow, they are further processed in the thymus. But B lymphocytes, they actually getting processed in the bone marrow itself. Pre-processing occurs in liver during the mid-fetal life, and in late fetal life and after birth, it takes place in the bone marrow only. B lymphocytes are different from T lymphocytes in two ways. One, they secrete reactive agents called antibodies. But in case of T cells, the whole cell develops the reactivity against the antigen. They are not releasing any proteins like your B cells. They are specific reactivity is shown by the T lymphocytes against the antigen. B lymphocytes have greater diversity than T lymphocyte, thus forming million types of B lymphocyte antibodies with different specific reactivities. They lodge in lymphoid tissue near the T lymphocyte areas. The B cells differentiate into plasma cells and memory B cells, and these plasma cells are the one who produce antibodies. So you can see in this diagram, this is the thymus, this is the bone marrow. So stem cells in this bone marrow give rise to lymphocyte precursors. We can now call them as lymphocyte precursors. They will be released into the circulation through the blood. They reaches the thymus. They are processed in the thymus to become the mature T cells, and again they are released from the thymus into the blood Through the blood circulation, they reach the lymphoid tissue. Here, the lymphoid tissue shown as lymph node. So, it can be lymph node, it can be spleen, tonsil, any lymphoid tissue. So, this is about T lymphocytes. And B lymphocytes, actually, these are the lymphocyte precursors. In the bone marrow itself, they become the mature B cells. They are released into the circulation. Through the blood, they reaches the lymph node, which is the lymphoid tissue, and they are lodged in the lymph node next to the T lymphocytes. So, the B lymphocytes and T lymphocytes, after become matured, they will be staying in the lymphoid tissue until their function.